Hello, my name is CoutureL, and today I'll be going over my main research project, the effects of subnatural background radiation on yeast cells within SNOLAB, a deep underground laboratory. I have no relationship with for-profit or non-profit organizations. This research project has received funding from NSERC, Bruce Power, the Nuclear Innovation Institute, and the Canadian Space Agency. Although high levels of ionizing radiation leads to negative biological effects, increasing research demonstrates that low levels of radiation can be beneficial, increasing cellular defense mechanisms. Low-dose radiation exposure includes natural background ionizing radiation, or NBR. While NBR is as abundant as air, surprisingly little is known about it. To understand the biological effects of NBR, we remove background radiation and examine how it affects cells. The two yeast strains utilized were wild-type and Rad51. The second strain is radiation sensitive due to having a deletion in the Rad51 gene, important for DNA repair. Each treatment condition was exposed to different radiation shielding mechanisms, leading to different radiation levels. The control was located on surface and exposed to the highest level of radiation, while the sub-background had the lowest radiation level and performed at a 550-fold reduction in background radiation. Radiation dose rate did not demonstrate significant effects on wild-type growth. Conversely, in Rad51, the background radiation dose rate had significant effects on doubling time. The sub-background cells consistently had the slowest doubling time. In terms of survival, both strains were not clearly affected by background radiation dose rate. However, the wild-type strain had much higher survival incidence compared to the Rad51 strain. Although the Rad51 strain background radiation dose rate did not have clear effects on survival, it did have significant effects on growth. This is likely due to increased DNA damage associated with a deletion in the Rad51 gene. Increasing research in this field enables us to implement more accurate radiation models for human safety and to better understand NBR. In this experiment, sub-background radiation was found to impact biological processes, indicating that NBR may be important for normal cell functions, and its absence may have a negative effect.